So many of you may be familiar with President Obama's Precision Medicine Initiative, which he announced for the first time in his State of the Union address a year and a half ago, and has been going forward since then. And its mission was to enable a new era of medicine through research, technology, and policies that empower patients, researchers, and providers to work together towards development of individualized care. And there are several agencies within the Department of Health and Human Services, as well as the White House, who were involved in taking up the different projects that are intended to move this forward. So FDA has, of course, a regulatory role in the Precision Medicine Initiative, and that was to optimize the regulatory oversight of next-generation sequencing. And we recognize that next-generation sequencing is already starting to lead to a lot of personalization within medicine, especially in the somatic space. It's also helping us to discover the basis for diseases that we never knew before. Because you can look at somebody's whole genome, you don't have to know what you're looking for, and you can identify what might be different. So our goal was to help ensure the accuracy of genetic tests, in this case specifically next-generation sequencing tests, and to develop an approach that's suited to the unique nature of next-generation sequencing. As you're all aware, next-generation sequencing can yield millions of results, if you think of a result in terms of a base. And it has the property right now of being rapidly evolving, both on the technological front, on the informatics front, and the clinical information that we're getting out of it is just pouring in. So we've got more information than we've ever had about people's genomes. And then the approach that we develop has to be adapted to, has to be put into our regulatory processes so they will be adapted so that we can do two things that they're slightly in tension with each other often, and that is encourage innovation while at the same time assure safety and effectiveness of these devices. And so we tried to find the sweet spot in the middle of this that would allow us to do both. So in the past, the development, maybe I will use the pointer here. Is the pointer here? That? Yeah. In vitro diagnostics and a lot of devices in the past have developed sort of in a, what I like to call, punctuated equilibrium sort of way, in which a technology was developed and came through FDA, and then it would sort of remain static for a number of years until the next iteration came along, and then you would come back to the FDA and you'd get another approval and so on. And the actual progress of technology wasn't especially fast. With next-generation sequencing and truthfully other technologies that are not genetic in nature, for example, proteomics, we're seeing that there's constant iteration. The technology is evolving super fast, and the knowledge that comes out of that technology is being aggregated and is also evolving super fast. So what we needed to do was figure out how to get from here to here in a regulatory sense so that the technology and the knowledge could evolve as they normally would and still be within regulatory bounds. So FDA generally looks at certain things when it's doing pre-market review of an in vitro diagnostic device. Our bar is safety and effectiveness. When you think about safety and effectiveness as applied to an in vitro diagnostic device, it's a little puzzling to think about, well, which part is safety and which part is effectiveness. So what we actually look at, we interpret that as, is the test analytically valid? And that is, does the test correctly detect the analytes or the substances that you intend it to? Does it do so precisely so that if you measure over and over and over again, you're getting similar results? And often, what are the limits of detection or the limits of measurement when that actually establishes your cutoff, your clinically significant cutoff? For clinical validity, importantly, we need to know whether the test correctly identifies the disease or condition or risk or whatever the output of the test is. And that's often expressed in terms of clinical sensitivity or specificity. more often in genetics now in terms of predictive values because there's a prevalence issue in genetics that has to be addressed. And in establishing clinical validity and analytical validity, the evidence must be scientifically valid, and we define what valid scientific evidence is in our regulations. We also look at labeling for the device or test, and what we're looking for are the directions clear, and is what you're saying about your test and what it can do truthful and not misleading? Is it supported by the data that are in your submission? And all of these three things that I've listed here are all based on what the exact intended use of your test is. So your analytical validation for one type of test might be different than it would be for another type. Clinical validity will be tied to what you say the test can be used for in the labeling as well. So we actually started out a number of years ago talking to a number of next-generation sequencing technology developers, not necessarily providers in the beginning, but technology developers, about what their technology did, how it worked, what the unique aspects of it were, what its possible error profiles could be. And we did a survey of that industry, just understand what we were starting off with technologically. And then we started going out and talking to laboratories and talking to test developers about how they were actually using these tests and what the issues were that they had identified and how these tests actually worked in their hands and what the benefits of them were and maybe what the downsides were. So we talked to a number of professionals through professional societies and so on, got a lot of information, did some surveys that informed us, and basically we came up with these two draft guidances that address analytical and clinical validity in very different ways than FDA has sort of done before, if you're familiar with the way that FDA has regulated. And the purpose of the draft guidances is to anticipate and be behind the needs of the rapidly evolving technologies. So we're trying to put in place a system that actually allows that evolution, which is generally a good thing, to happen at its natural pace. Of course, as FDA, we want to support reliable, accurate, and understandable test results. That's sort of fundamental to our mission. And under the PMI, we were very cognizant of the need to promote an efficient path to market for these kinds of tests, for all kinds of test developers. We're not picky about who's developing the test. We're talking about the technology and how to get tests to market here. And in order to promote that efficient path, we thought through what do we really probably need here. And one of them is we really need groups or companies, as Trevor said, or others to develop and implement standards that help to assure test quality. Now, these can be methodology standards, like how to do it. They can be material standards that actually have a specific value. There's all kinds of standards, and they're all useful here. We also wanted to describe, I think, a fairly unique regulatory pathway for NGS-based tests. And we have limited in the beginning to a particular intended use, and that is for diagnosing heritable genetic diseases. And the reason a lot of people want to know, how'd you pick that one out? We picked one that we thought we understood well and that we could generate principles around, get comment and feedback, so that we would have a baseline from which to work to add in other intended uses. And I'll talk more about that in a minute. And we also wanted this to be based, as we move forward in FDA, we're doing more and more of this based on open processes and accessibility. We want transparency. We want people to see what we're doing, and we want people to show others what they're doing, because we've found, especially in our bioinformatics work using Precision FDA that some of you may know about, this sort of open source, transparent work really allows other people to look at what you're doing and figure out if something's not going right, how to fix that, and then everybody gets the advantage of the fix. It's a little bit maybe not the way businesses have been run in the past, but we think that this would enable more rapid good use of next generation sequencing importantly. So the really critical thing here is I'm talking about draft guidances. For those of you who are not familiar with the guidance process, we put out what are called draft guidances that articulate our best thoughts at the time, and then we leave a comment period open. In this case, I think it's 90 days, where we expect the public to read the draft guidances and comment back to us and tell us what about the guidances you support, what about the guidances you think is wrong, is there something missing, did we address something that's not relevant, and for the community to tell us, you know, this is the way it should look. And then we take all of those comments, address them, and publish a final guidance, which usually then is considered to represent FDA's current thinking. Guidances aren't binding, but they do represent our thinking. So if you decide to do something that's not described in a guidance, then you usually have to talk to us about it. So let me start with the first draft guidance, and that's use of standards in FDA regulatory oversight of next-generation sequencing-based in vitro diagnostic used for diagnosing germline diseases. So this is, we call this the analytical guidance, and it talks about analytical standards. And as I mentioned before, NGS tests, we realize, can have a number of broad intended uses. If you take all the possible intended uses and put them into buckets, they include things like for diagnosing infectious diseases, for non-invasive prenatal testing, for risk of disease, and so on. And as I mentioned, we decided to start am I pointing? Oh, okay. With hereditary disorders, because we thought that was fairly well understood. People were doing these tests, and we were able to get a lot of information out front. So the draft guidance actually only applies to this particular intended use right now, but the principles will probably be more broadly applicable, and we will move to other intended uses in the future. So this guidance provides some technical recommendation, and it describes a specific approach to test design, which is something that you don't necessarily always see in FDA guidances, because design is something that is generally covered in the quality system regulations, but we really want people to think through as they're designing these tests what they're designing them for and how they're going to validate them. So we do describe the approach to design. And we believe that our approach accommodates a lot of different test designs. So it's not just one way. You can do it a bunch of different ways. It just needs to be within sort of the big container of test designs. You can use different components. You can have slightly different indications as long as they fit in this big bucket. And what we have recommended here within this guidance, although they are recommendations, what we'd really like is for a standards developing organization to turn them into standards, like ISO standards or CLSI standards or something that FDA can then recognize. So why do we want that? Why doesn't FDA just want to say, well, this is how you should do it, and just let FDA say it? As science changes, as technology changes and so on, you need to change your recommendations over time to keep up with it. And FDA's ability to do that through regulation is actually kind of slow and administratively intense. So we have to go through rulemaking and all of these processes, and it takes a long time. And sometimes by the time we're finished, what we're talking about is past. It's not so relevant anymore. Standards organizations can do this much more quickly. They can revise a standard rapidly and so on. And the important thing about standards developing organizations, not in comparison to FDA, but maybe in comparison to some of the other things you think about, is they're required to have a consensus. So they have to take the opinion of all the stakeholders and consider it. And the output of that, a standard, has to be a consensus document. It can't just represent one sector or another sector. It has to be everybody coming together. And then FDA can recognize that standard as we agree with that, and yes, you can go ahead and do that. So our design standards start out with this concept of test design and move the arrows sort of go off in two directions, but these things can just happen at the same time. The test design will drive the test performance and the test run quality metrics. You will then need to actually assemble the test from the various components and do the validation. And when you do the validation, you may actually find, oh, this doesn't work exactly the way I wanted it to. And often that's because your design parameters in the beginning weren't exactly quite right. So you go back to the design phase and fix whatever needs fixing, and then move through this again. And it can be an iterative process until you've actually got the test you want that works the way that you want. And that's what our recommendation here centers around. So the test design is generally informed by intended use and user needs. So you know what you're offering the test for. You're going to go out and ask clinicians, what do you need? You're going to figure out what your market is and so on. And you're going to figure out what the user actually wants from this test. And the user can be the laboratory. It can also be the person who gets the results. You're going to have to decide what kind of specimens are appropriate for testing in your test. You usually need to do that up front so that you can design the test properly. And for NGS, you need to understand what regions are you going to interrogate to get the answers that you want. Once you have that in place, you determine what the performance needs are, how accurate does it need to be, how precise, what is the limit of detection and so on. And I can't even see that one at the end now. I don't know what I did with my glasses. Anyway, I can't read that, I'm sorry. Okay. And then you decide, yes, what test components and methods you're going to need. I'm sorry, I don't give this presentation very often. But yeah, so that you then select the test components and methods that you'll need in order to achieve the design that you want. And we're well aware at FDA that there are a number of vendors of all the different components that you could possibly use: the library prep, the instrumentation, the reagents, the informatics pipelines, different pieces of the informatics pipelines. And then at the end, there may be interpretation software. The interpretation may be done by a human. We sort of understand that whole big picture. In order to create the test you want, you're going to have to figure out what the performance characteristics are that you need and identify the performance metrics and set what are the minimally acceptable values. And this can change depending on what you're detecting and why and what else is out there. Is this an improvement over nothing? Is this an improvement over an existing test? Blah, blah, blah. And the guidance specifies 4 overall performance metrics that we think, again, this is draft guidance and you get to come back and tell us if we're wrong, we think should always be assessed, and that is the accuracy. How well does it actually detect the thing you want it to detect? Precision in terms of reproducibility and repeatability, and that is if you run the same thing over and over and over again, under the same conditions or under change conditions, like with different operators, different days, maybe different instruments and so on, are you always getting sort of the same result? It's often important to look at limit of detection because that's often the cutoff between clinically, yes, actionable or not actionable. And then the analytical specificities. You need to make sure that the stuff you're putting into the test isn't actually causing the test to go off the rails and misreport. So we look at cross-reactivity and nucleic acids, because a lot of nucleic acids can cross-react with other nucleic acids because there's only 4 bases in the genome and they hybridize. So we worry about things like pseudogenes and things that look a lot alike. And then cross-contamination. Interference is usually not a big deal, although it could be in some areas where you get a substance in there that actually prevents the reaction from working properly. So these are the kind of performance characteristics we're thinking are important. The minimum performance thresholds, as I said a minute ago, this may actually vary according to what you're actually trying to do. We generally stayed away from specific numbers here. We did put in, we thought the accuracy should be very high, and we thought 99.9% sounded like a good number. And we talked to a lot of people around, and they said, yeah, that was good. And then the precision, that is when you run the test over and over in the same and changed conditions, that the lower bound of the 95% confidence interval for precision should be 95% or greater. Now, I know a lot of people are going to come back and say, you know, maybe for my test that's not possible or whatever. And again, this is guidance. If you can justify something different, that we're always willing to talk about it, and we generally accept it if it's well justified. We talked about test run quality metrics. We realize that different developers use different methods. We basically need to know whether you should accept the result that's coming out of your machine or your pipeline. And we specified coverage as the only metric that we think you should always use. Not sure that should always be used or that's the only metric, so we'd be looking for comment on that. And we expect the test developer to select other metrics that are relevant to their test. And we're generally looking at end-to-end testing here. We're looking at a test system. So we also provide general recommendations for performance evaluation studies. These are analytical validation studies. You might also have heard of them. And we really do need to see end-to-end performance here, and that means start with the specimen and give us an answer so we understand how the whole test system works. And even if you just want to bring forward a component, it always has to be evaluated within a test system if it's actually part of the test system. So there are various performance aspects. You need to look at representative genomic regions, variant types, sequence contacts. I think Trevor just told you earlier that in some FASTQ files that were sent around, people got the SNVs routinely, but they missed when it came to indels. So you need to actually identify how well you do on all the types of changes. It's always good to know what kinds of variants you actually can't detect very accurately. And so if you get that result, you look at it and you ask yourself, is that really true? Do I need to check it with another method, or does it correspond with the clinical presentation of this person? And then you need to use the appropriate specimen types. I know this is very difficult in next-generation sequencing. A lot of these specimens are hard to come by if you're doing something in rare diseases, if you're doing something in rare cancer. And if you actually can't use a natural specimen and you're using something contrived, you need to understand the commutability, how much of the variability in the natural specimen is represented in that contrived sample, because a lot of times it can make a huge difference. And then we leave it up to you, and this has always been true, up to you how many specimens you need for your validation experiments. So when we say it's up to you, the catch on that is you have to convince us that that's the right number. So we're often looking at statistical figures here, is how much confidence will you have with a given number of specimens, and how many wrong results can you get and still have that confidence, and so on. So I encourage you to think about it in scientific terms and not necessarily just what's convenient, maybe in the freezer or what you can buy and so on. Accuracy studies, we're looking at accuracy in comparison to a comparator method. And to a greater degree now that we have Precision FDA and a lot of sort of reference data sets out there, not so much the method, but the test-generated sequence in comparison to sort of a reference database data set. There are a number of them that are available, and if you've been participating in Precision FDA, there are a number of them there that you can use, and you actually compare your sequence output to the known truth of these reference data sets, and you can see what you've gotten wrong and what you've got right. And then the important thing, I think, perhaps more for labs than for customary IVD manufacturers, is documenting the results of your validation. I'm sure most labs do this. I'm sure it's expected, but actually writing down what you did and why you did it is really important to FDA. And we need to understand things like confidence intervals. We need to know by variant sequence context and so on. But the critical thing is that you write it down. And document it in some way that you can show someone else. We talk about a little bit other recommendations, like do you need to follow up a positive with another method? More and more we're thinking maybe you don't. Next-gen sequencing is actually getting pretty good, although there are still areas where our informatics are not as good as we'd like. how you do your variant annotation and filtering, how you present the test's performance to others, what the test reports look like to some degree. FDA only will look at the part that relates to your claims. It doesn't look at the practice of medicine part in which I, the physician, am deciding what this result means for my patient. It looks at things related to your claims. And then, of course, how you're going to deal with modifications. You're going to change your test over time. Sort of how are you going to, you're going to have to revalidate the entire test, or are there only certain steps you have to do, and so on. So probably one of the most interesting things here for many of the stakeholders is the regulatory considerations in this guidance, where we talk about if we haven't classified a test before or a particular intended use before, by default, under the law, it goes to class 3, and that's the generally considered high risk, but it's actually not completely risk-based. So if we haven't seen it before, by default, it's class 3. What we can do is to down-classify these tests out of class 3 into a lower class through an administrative procedure called a de novo. And we looked at this intended use for looking at heritable genetic disorders, and we said, you know, we can probably think about that as class 2, or sort of moderate risk, where general controls and special controls would be enough to control the risk. And so we've actually said that in a guidance. We almost never say ahead of time what we think classification would be. And then we even discussed the possibility that in the future, if we had all the tools in place, that these tests could be exempted from pre-market review. And this is just the timeline by which that would happen. It would start out as class 2, go through de novo, get down classified, and then be exempted. So let me go on to the next guidance, which is about the human genetic variant databases to support clinical validity. We call this the database guidance. And we think we should be moving more in this direction as a society, where we're aggregating evidence so that especially rare things and even not rare things, we get all the evidence in one place rather than having it siloed all over the place. And the advantage of this is clearly evidence is generated by multiple parties. And as you aggregate evidence, you get a stronger evidence base. And it's often close to the current state of scientific knowledge. And as it improves, you may start off saying, we're not sure what this means. And as you get more and more evidence, you may be able to say, oh, that change is benign, that change is pathologic, whatever. So we think this is a way to keep up with evolving evidence through databases. Our draft guidance deals with publicly accessible databases only. That is, it has to be accessible to anybody who wants to use it. We make recommendations to the administrators of these databases of how they can set up their database so that FDA can consider the information in there to be valid scientific evidence. And if you remember at the beginning of the talk, I said that's clinical validity relies on valid scientific evidence as defined by FDA. This pathway is entirely voluntary. We're not regulating databases, we're recognizing them, and that's a completely voluntary process. And we go on to say that we think the evidence from these databases could support clinical validity. So that means you, as the test developer, don't actually have to go out and do clinical studies. You might be able to point to a database and say, all the evidence for what I want to claim is right there. And if we've recognized the database, we can say, okay, that's enough, or we can say that's good, but we need a little more, and so on. So our proposed recognition of a database, remember this is not regulation, as it would be voluntary. We've laid out a number of policies and procedures that we think are important in order for us to recognize a database. And we think it's important to actually maintain this recognition through perhaps annual relooks at what the database is doing so that it doesn't drift off in a direction that we wouldn't be happy with. So what we're saying is that the data and assertions from databases that we recognize would generate valid scientific evidence and would constitute valid scientific evidence in most cases that we could recognize this supports your claim. And we talk about various ways the assertions have to be presented. They can't be false or misleading. They should be descriptive and so on. So I think that guidance is a little more on the face of it straightforward, but what we're trying to do is to use a buzzword, crowdsource evidence, so that every test developer doesn't have to go out and do their own clinical study, and that we get more power and evidence more rapidly by aggregating data. So in summary, the guidances are intended to encourage innovation by allowing the test developer to incorporate new things in test design, new clinical information rapidly, while at the same time assuring the quality and reliability of the test. And we'd like to promote adoption of these tests into the community if they're good tests, because we think this will really support the advance of precision medicine. We've tried to assure that the test development according to these guidances would have an efficient path to market. That is, we would really compress the timeline between development and first offering, and that we might even exempt these from pre-market review and use third parties actually to assess the conformity of a lab to the standards that we hope you'll help us develop. Our next steps are that the public comment period is open. I think it closes on October 6th. 90 days, which is a little longer than we usually give. And if you look on our website, which I'll give you next, that you can find the FR notices or how you comment, tell you how to comment. We will be having a public meeting on September 23rd that requires registration. So if you go to our website, you can register for that. It will be held at NIH here in Bethesda. And we published the FR notice, I think on Monday, and we've already got over 125 registrants, so don't drag your feet, because there's only so many people we can accommodate, and we will have a separate docket for comments to that meeting. And then finally, I really just want to encourage you to submit comments to us. We've tried to make this an inclusive, community-driven type of process. We want to maintain that through the commenting process, and we really encourage you to give us substantive comments, like what you think we got wrong, what you think we got right, did we miss something, did we include something that wasn't important, in a way that we could actually read that and say, should we change the guidance? And we will be needing your help. If you are a member of a standards development organization or want to be one, we encourage you to present this as a possibility for standards development that FDA could recognize. We need more reference materials and physical standards that can move forward. So we encourage anybody who's in that area to keep going, to share data, to work with sustainable, high-quality databases, and to help out with curation. This is going to be on an expert basis. It actually takes people to curate and to make assertions. So any of you who are in that area, we really would love you to be helping with these databases. And then if you have questions about any of this, you can email us at this address. And this is our website that should have all the information that is publicly available about what we're doing.